Lesson 9 will be a little bit different because we're really going to summarize what's happening within the body in terms of training adaptations. And then it's really the last lesson of its type because lesson 10, or your final lesson, will be a summary taking everything that we've learned and pulling it together to generate an eating strategy or plan for our client. But studying training adaptations is one way to summarize what is occurring in the body in terms of metabolism when we train our client in a fitness environment. Let's go ahead and look further at some of these changes. Lesson 9 outcomes include being able to describe the main adaptations to resistance and endurance types of training. Then we want to be able to discuss the mechanisms behind these changes and some of the molecular mechanisms that underlie these changes. Furthermore, we want to discuss how carbohydrate intake can influence signaling, protein synthesis, and training adaptations. Students should be generally aware of how antioxidants can influence signaling, protein synthesis, and training adaptations. And lastly, understand how carbohydrate intake can reduce symptoms of overreaching. Protein synthesis is regulated by signal transduction pathways that sense and compute local and systemic signals and then regulate various cellular functions. The main signaling mechanisms are responsible for the growth, metabolism, and proliferation of muscle fibers and most other functions depending on signals like nutrient availability, pH, the partial pressure of oxygen, reactive oxygen, and even mechanical stimuli. The nervous and endocrine systems also provide further inputs, and cells detect this constant changing mix of signals through specific sensor proteins of their own. During exercise, cellular homeostasis is disturbed and initiates a cascade of events that ultimately result in an adaptation that will cause less disturbance of homeostasis the next time that the same exercise is performed. This is another way of saying that the body is adapting. Now, during exercise, these signals include changes in muscle stretch and muscle tension, as well as changes in intracellular calcium ion concentration, changes in the energy charge of the cell or its polarity, and changes of the reduction potential. Making things a little more complex, certain hormones will then bind to receptors on the cell surface, and this also can alter these signals. These primary messengers can then trigger a series of secondary molecular events that refine translation or refine transcription of the event occurring in the body. And this is another way that the body adapts. Now, most of you know that during muscle contraction, ATP is broken down to provide energy, and in this process, ADP is formed. ADP is then resynthesized to ATP by glycolysis or by oxidative phosphorylation. Some ADP is further broken down to AMP. The ratio of metabolites ADP, AMP, and phosphate ions relative to that of ATP is often referred to as the energy charge. Now, if there is a lot of ATP present and little of the metabolites, the energy charge is considered high. If the concentration of metabolites rises, the energy charge is now low. These metabolites are important regulators of metabolism, but they also serve as signaling molecules. Now, we already have learned about the importance of carbohydrate replenishment before, during, and after exercise, but it's been suggested that carbohydrate intake during exercise might actually interfere with training adaptations. And this idea or theory is based on the observation that carbohydrate intake during exercise can reduce the expression of the messenger RNA of certain proteins after exercise. So while research or studies will continue to show conflicting results about this messenger RNA, it seems unlikely with what we already do know about carbohydrate replenishment and how it would really not seem likely to have a negative influence on training adaptations. Like many topics in our field, before we could say with any certainty that there's a relationship between carbohydrate intake and messenger RNA, we would need more study and research on the topic. Now, we've learned a fair amount about glycogen levels, but decreased glycogen levels can result in disturbances of the endocrine environment. Glycogen depletion is related to high levels of epinephrine and norepinephrine, cortisol, and glucagon, while insulin levels are very low. These hormonal responses will result in changes in substrate mobilization and utilization. For instance, high epinephrine levels in combination with low insulin levels will increase lipolysis and stimulate the release of fatty acids. These hormonal responses are generally good to understand because they can be an influencing factor in your client's body fat storage patterns. Branched-chain amino acids, in particular leucine, are signaling molecules as well as building blocks for protein synthesis. And although the branched-chain amino acids alone can stimulate signals within the muscle, this increased signaling will not necessarily result in an increased rate of synthesis if other amino acids required are not available in the blood. Now, many of your clients might be taking antioxidant supplements, but antioxidant supplementation may decrease training efficiency by preventing some cellular adaptations to physical activity. And although we know a high carbohydrate diet can maintain muscle glycogen stores and reduce or delay symptoms of overreaching during prolonged periods of intense training, overreaching cannot be prevented by a high carbohydrate intake alone. The benefit for coaches or trainers in knowing basic training adaptations is that you know you're on track if your client is adapting properly to exercise or changes in their food intake. As part of a macro assessment, you might analyze 2 to 3 months of food logs for a client once they have reached their goal weight. As more of a micro assessment, you might actually look at food logs from your client 3 or more times per week if you're working with the client at that level of intensity or trying to get biometrics under control.